Contrary to what you might have seen in the movies, cloning is not about making copies of people or animals, but rather it's about making recombinant DNA that's able to self-perpetuate. So let's say you have some DNA, but there's one part that you're interested in and you want to either store it or make copies of it or just maintain it for future use. What you'll need to do is create a piece of DNA, usually circular DNA or a plasmid, which contains your DNA of interest, which you can put into a living organism such as a bacterium, and it will replicate as the organism replicates. For this to work, you need to have some other things in your plasmid. So first of all, it needs to have an origin of replication, since otherwise it won't be able to replicate when the cell divides. What else do we need in the plasmid? Well, one thing we have to be concerned about is that some bacteria might lose the plasmid, and over time those bacteria might take over the culture. So we want to make sure that only bacteria with our plasmid are able to proliferate. We can do this by adding an antibiotic resistance gene to our plasmid, and then we'll only grow the bacteria in media that has an antibiotic. So any bacteria without our plasmid will die. So how do we put this all together? The first step is to isolate the DNA you're trying to clone. One way to do this is by cutting the DNA upstream and downstream from the site you're interested in, using enzymes called restriction enzymes, which can cut the DNA at specific sequences. If the DNA you're interested in happens to be a gene, you can also collect the RNA you're interested in and reverse transcribe it to make the corresponding cDNA. So what's the difference between a cDNA and the original DNA sequence? Since cDNA is made from mRNA, it has no introns. So now we have our DNA of interest. Next we need a plasmid. Fortunately, we're in the 21st century, and you can just buy a plasmid that already has everything you want in it, like antibiotic resistance regions and the origin of replication, and a bunch of different sites that are targeted by different restriction enzymes, which you can use to cut open the plasmid and insert your sequence. As a flashback to our blotting procedures a few slides ago, do you remember which blotting procedure would allow you to determine which DNA sequence or restriction enzyme can bind to? That would be the southwestern blot, which is used to detect DNA binding proteins. So first, you'll use a restriction enzyme to cut open your plasmid, and then add your DNA of interest to the mix. And what enzyme do you think we can use to seal them together? This would be ligase, just like in regular DNA replication. Then you can use a process called transformation to get this plasmid into a bacteria and then let them divide. So what's the point of cloning? Well, one purpose is to amplify DNA, just like in PCR, which I talked about earlier. So if you clone DNA into bacteria and then let them do what they do best, which is divide, you end up with a lot more copies of DNA than what you started with. Another use is to make a library of all the genes which are expressed in your sample, which is called a cDNA library. In this case, you're not cloning a specific sequence, but instead you're reverse transcribing all of the mRNAs that are present into DNA, and then each bacterium will get one, so your resulting population of bacteria will contain a library of DNA corresponding to all the genes that were expressed. So in addition to your DNA of interest, what two features should be on a plasmid you use to clone DNA? This would be an origin of replication, since without it the plasmid won't replicate along with the rest of the DNA in the cell, and an antibiotic resistance gene, so that you can select for cells that have your plasmid in them.